It's a pleasure to be here this morning, and I appreciate the opportunity the committee has given me to share with you my perspective about this really serious problem, both of inadequate treatment of pain and of the opioid abuse and opioid overdose that we see as a societal catastrophe. From my perspective, as I heard the eloquent discussion of the first panel describing the problem, we have an enormous fire which we are trying to put out with inadequate tools, and frankly, with what the means we have at the moment are inadequate, and the fire is not going to go away. There is a solution, and that is we need to develop new drugs, new analgesics that are as effective or ideally even more effective than opioids without their abuse liability. And that is the solution. That is the key point I want to make. But that is not going to come out of nowhere. That's going to require a massive investment, both by the federal government and by industry, something that has been lacking to date. Nevertheless, I'm extremely optimistic that the state of our knowledge of pain mechanisms has reached a point at which this is a realistic alternative, so that while we struggle at the moment to deal with patients who are inadequately treated and we try and deal with the opioid crisis, we need to keep our eyes ahead of us for the near future by investing in the development of new effective treatments. So the identification of targets for new analgesics is contingent on understanding the neurobiology of pain. Without that, there are no simple ways by which we can get a single drug that will be effective for all patients. And the insight that we've gained over the last few years is that by just treating pain as a single entity is both inadequate and incorrect. I'll illustrate this by looking at neuropathic pain. The standard clinical approach to this is to try and identify the pathological cause in a patient, the anatomical location of some pathology, whether it be nerve injury or some metabolic or infectious condition, which then produces a clinical syndrome that we can identify, such as diabetic neuropathy or HIV neuropathy, that generates a series of symptoms, the pain, both spontaneous and evoked, and based on that, physicians choose a treatment, which, as we recognize, are generally inadequate, and in particular, with a major problem, we do have no tools at the moment to identify who will respond and who will not respond to any particular treatment. What the growing realization that this model is not the best approach to the management of pain has come from our appreciation that the drivers of pain reflect the mechanisms in the nervous system that are producing the pain in the patient. And these are diverse and complex and vary from one patient to the other. So yes, we certainly have the pathological, etiological factors, but they operate in the context of the unique genome of each of us, and that the genetic variance is a major factor in why one patient after surgical damage to a nerve may have no pain whatsoever, whereas another patient may have very severe pain, why one patient who's exposed to a cancer chemotherapeutic agent may have no peripheral neuropathy, whereas another with the equal dose may have severe pain of such intensity that they are unable to complete their therapy. And these are largely, these are a combination of environmental and genetic factors, but we have not invested enough in identifying what these genetic variances are. We recognize from the progress that has been made in psychiatry that we need literally hundreds of thousands of patients who need to be sequenced to identify the genetic variants that are contributing to this. We also now recognize that many of the pathological mechanisms that operate as a consequence of these pathologies, and I just illustrate in this diagram some of these. There may be ectopic activity in injured nerve fibers. There may be a change in the threshold of activation of nociceptors, and there may be altered brain connectivity. And the challenge for us is to be able to identify what these mechanisms are, because this is going to be the basis in which we can make a precision medicine approach really work. In other words, what are the drivers in the patient who's sitting in front of Jim Rathwell that are causing their pain, so that he can select the treatment that will target that particular mechanism. And I would strongly argue this is the way that we need to see this problem. I'll illustrate this by looking at just one of the symptoms of pain that patients may have. And this is heat pain hypersensitivity. We all normally have a threshold for the differentiation of a warmth stimulus to a painful stimulus at around about 42 degrees, but we're all familiar with, after sunburn, this drops such that a normally warm stimulus now becomes unpleasantly hot. Patients may have similar heat pain hypersensitivity, and as we've dissected out the molecular drivers and mechanisms of heat pain and the changes that can occur in the neuronal apparatus that mediates that, we have identified multiple components to this. These may include the sensitization of the nociceptors by inflammatory mediators, changes in the excitability of the nociceptors, particular polymorphisms in some of the transduction channels like TRPV1, which alter their sensitivity, and the presence of endogenous agonists that modify these nociceptors. And the reason I highlight this is that each of these includes different targets, and I've just labeled some of them. For example, COX-2 with the production of prostanoids, which then act on EP receptors. Excitability being driven by changes in sodium channels or potassium channels. The sensitization being mediated by intracellular kinase pathways that lead to post-translational modification of TRP channels. Each of those requires a different treatment, and in order for that to be effective, we need to be able to know in our individual patient which of these pathways is contributing to their pain hypersensitivity. So one of the challenges is in finding novel analgesic targets, and I'm not going to, that's an enormous topic which I can't really discuss in great detail here. Progress has been made. I just illustrate here some of the targets that are being actively developed at the moment, including selective sodium channel blockers, TRP and voltage-gated calcium channel antagonists, TRAC-A, which is the receptor for NGF as another target, and potassium channel openers, and indicate some of the drugs that are either in preclinical or undergoing clinical trials at the moment. So one of our challenges then is to identify targets that are contributing to the pain mechanisms that are present in our patients, and then we can develop treatments to affect those particular targets. We need to recognize the complexity of the biology, though, in that the old approach that a single target operating by itself is enough. We can then either activate or block that target in some simple way using a screening platform where typically a large pharmaceutical company would throw 3 to 4 million compounds against a heterologously expressed target and find something that uniquely acts on that target is no longer enough, because the target biology is much more complicated than that. For example, we now recognize that GPCRs, the G protein coupled receptors, can affect different kinds of signaling, and that biased ligands to these GPCRs enable us to determine one form of signaling and not another. And this, in the case of mu-opioid receptors, offers us the possibility of producing a reduction in pain with a reduction in the risk of adverse effects. There's also a recognition that the these G protein coupled receptors may form homo and heterodimers, and that the properties of the heterodimers may be very different from the homodimers, and these require a different kind of pharmacology. And again, expressing a single target by itself will not find drugs that act specifically on these heterodimers. And even furthermore, there are coupling between G protein coupled receptors and ion channels, which again represent another alternative strategy. So again, we need a massive investment in understanding how the molecular biology underlying pain mechanisms, and that frankly is something that has been missing, and until that investment occurs, we are not going to make the progress that is going to transform our approach to the management of pain. So these are some of the major targets that are being actively developed. I think Jim Rathmell put it very clearly that at present there are no new, in the last 5 years, there have been no new analgesics acting on novel targets. We've been basically playing with the standard targets, the mu-opiate receptor, by using reformulation and frankly substituting one problem with another, until we get true novel targets which operate in ways that are distinct from the opioid receptor system, which Dr. Fields will be telling us about. We are going to really struggle. But you know, it is clear that there are many potential targets, and I just illustrate those here. And these are ones based on an analysis of activity in the pharmaceutical industry that are looking at these. So we have to have a clear idea of what we're looking for, and this is the kind of analgesic checklist that I'm sure we'd all agree with. We need a drug that acts on a target that will attenuate pain symptoms, and the clinical impact of such treatment needs to be higher than we can currently manage. I think while integrative approaches are obviously fine, and particularly now at the moment when we don't have alternatives, unless the impact, the actual clinical significance, the amount of reduction in pain that we can produce is significant, we are just scratching on the surface. A key feature is we need to have drugs that do not cause major adverse effects. This has been mentioned. If our patients struggle to take the drugs because of the adverse effects, whether they be sedation or even more significantly the abuse liability, we are not going to make progress. So this has to be integral to our strategy to identify targets where these are not features of any drugs that will act on such targets. We need tools to identify how to validate whether particular targets are going to be useful. And one of the major problems that we struggle with in drug development is to make sure that when we give a drug to a patient, the drug is actually engaging with that target in a way that can impact on the pain. And we need markers of that, biomarkers of target engagement. And in many cases, the many failures in the development of novel analgesics have come from a failure to actually engage the targets. And so we abandon targets just simply because the drug that was administered has not engaged it. It was not that the target was the wrong target. And then, again, mention has been made, we need biomarkers of efficacy, and this is extremely difficult. I think the illustration by Dr. Thomas of of expecting a patient to record their fever by a visual analog scale of different levels of heat is spot on. Until we get biomarkers of pain that move beyond those kinds of subjective, descriptive we're going to struggle, because we need a quantitative assessment of how much pain our patients are experiencing and how much relief our interventions can impact upon that patient. And until we do that, we're in an arena where we are lacking the quantitative information to make to move forward in the most effective way. So, rather than the standard pathway, which is largely a case of trying to treat symptoms, regardless of how the pain is generated, what I'd like to argue that we need to recognize that our patients, each of our patients have different genotypes that may determine what kind of pain they have and how they may respond to different treatments. And to explore that, we need a major investment in understanding the genotypic drivers of both pain and response to treatment. We need to recognize that in each patient there may be an individual neuropathology, and we need to find out exactly what that is, and that too will require a major investment. We've heard in the first panel a definition of the need for research in this area, but the National Pain Strategy had almost nothing about research in the underlying mechanisms of pain, and that, frankly, is a great omission in my opinion. And until we actually are able to identify how changes in the nervous system actually produce pain, we are not going to be able to manage this in a rational and effective way. So that needs a massive investment, and it needs to be a top priority. It's not going to deliver immediately, but in the long term, that is going to be the solution. We need to be able to identify in patients exactly how their pain is being generated. So we need those kinds of clinical studies, and then we can result in a situation where we can have an individualized precision approach to pain management. And again, this is not unique to pain. This is an approach that we have to develop. If we look at the failures in drug development, to a large extent, they are based on failures in efficacy, and that is particularly true in pain. The FDA sees safety as a key factor, and of course that's important, but the failures in drug development are failures on efficacy, and that is particularly true in the case of the nervous system. And so I think we really have to push aggressively in trying to improve targets and that have efficacy. So I'd like to end up by just saying that as we look at drug development and our plans for novel analgesics, we need to realize that the way drug development is conducted at the moment, which is a serial process, starting with selection of a target, then looking for drugs that interact with the target, and then testing that in preclinical models, and then in looking at safety in phase 1, and then some efficacy signal in a proof of principle study in phase 2, etc. If the wrong target is selected, if the wrong preclinical models that are not predictive of efficacy in patients are used, then we have a sure failure. So what we need to do is look for new ways of identifying targets, and I won't go through all of these other than to say that there are new technologies, stem cell-based capacity to make human sensory neurons, human spinal cord neurons, human thalamic and cortical neurons, which we can study from patients who have pain, and this is going to provide us with a tool to do phenotypic screens in ways that have not been managed, we could not have imagined until very recently. This, combined with genome editing technology, is really going to transform our approach to identifying targets and seeking drugs that act on those targets. We need to transform the way we do preclinical studies. We need to move away from a very simple-minded approach where we look at reflexes as a surrogate of pain to ones that interrogate what an animal is feeling and how that changes in pathological conditions that mirror those conditions that occur in our patients, because that is the way we can test whether novel analgesics work or not. And then we need to study in our patients the genetic variants that contribute to the risk of developing pain and their response to different treatments. And that will include a detailed human pain phenotyping that goes beyond that simple subjective descriptors. So this is my final slide, which embraces the complexity of the problem that we're facing, that we can't see this just as a simple series operation. Our target, in order to choose the best target, we need insight from model organisms. We need insight from patient genetics, and that we need insight from the failures and successes that occur in the clinic. So we need a loop. We need to re-examine the way in which we identify novel compounds. As I've indicated, stem cell technology is certainly one way to do that. So the patient can be the driver of target selection. And then we need to transform the way that we actually measure the potential effectiveness of our novel analgesics in patients in a way that recognizes that there'll be responders and non-responders, and that the effects on the patients are much more complicated than just a simple reduction on a visual analog scale. Thank you.